Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. So, you know how our bodies are constantly building and repairing themselves?
Right, it's a non-stop process.
Well, for decades, scientists kind of thought this key repair crew for our DNA, an enzyme called telomerase, they figured it worked pretty much alone.
That was a standard picture, yes.
But get this, a groundbreaking new study has just revealed it's actually more like a tag team, a complex intertwined dimer.
Yeah, two units working together. And this discovery completely reshapes how we understand, well, really fundamental stuff like aging and cancer.
It's a huge shift, isn't it? Moving from picturing telomerase as this single actor to seeing it as a dynamic duo.
Exactly. And that changes everything about its role, how it functions, and maybe even how we think about designing new therapies down the road.
Absolutely. So today we're taking a deep dive into this exact finding. It comes straight from a recent paper in the journal Science.
The title is Cryo-EM Structure of Human Telomerase Dimer Reveals ACA RNP-mediated dimerization. Bit of a mouthful.
Huh? Yeah, just a bit. Our mission today is really to cut through that scientific density. We want to show you not just what they found, but really why it matters.
Why it's important for genome stability, the whole aging process, and of course, the fight against diseases like cancer.
And how this discovery is actually opening up some brand new avenues for potential treatments. We'll get to the core insights.
Okay, so to really appreciate why this is such a big deal, maybe we should quickly lay some groundwork.
Good idea. Let's talk basics. Telomeres.
Right, so imagine your DNA all packed up into chromosomes. At the very ends, you have these protective caps. Those are the telomeres.
Kind of like the little plastic tips on your shoelaces.
That's a perfect analogy, yeah. They stop the important genetic stuff, the shoelace itself, from fraying or getting damaged.
Okay, makes sense. But there's a problem with them, isn't there? Something about cell division.
There is. It's called the end replication problem. Basically, every single time one of your cells divides, the DNA copying machinery can't quite copy the very, very end.
Just a little bit gets lost each time.
Exactly. A tiny bit of that telomere cap gets clipped off, and if that just kept happening cycle after cycle.
Your telomeres would get shorter and shorter until, what, the cell just stops working properly?
Precisely. The cell eventually enters a state called senescence. It stops dividing, or it might even die. This is linked to cellular aging and broader tissue dysfunction.
Okay, so we need a way to fix that, to counteract the shortening.
And that's where telomerase comes in. It's this really remarkable enzyme, a molecular machine, actually.
What does it do?
Its job is to add those lost DNA bits back onto the ends of the telomeres. It's like a specialized repair crew just for those shoelace tips.
And it's made of?
It's a ribonuclear protein, or RNP. That means it's part protein, specifically a reverse transcriptase called TERT, and part RNA, which acts as the template for adding the DNA repeats. In humans, that RNA part is called HTR.
Got it. And this activity, it's pretty important then?
Oh, absolutely crucial. It's vital for things like stem cell renewal, allowing those cells to divide many times without losing their telomeres. It maintains genome stability overall.
But it's also involved in cancer. You mentioned that earlier.
Yes, that's the paradox. While it's essential for longevity in some cells, in over 85% of human cancers, telomerase gets switched back on inappropriately.
Letting cancer cells divide indefinitely.
Oh, exactly. It makes them essentially immortal, overcoming that natural limit to cell division. So it's vital, but also has this dark side and disease.
Okay. So, this super important enzyme, there was some debate about what it actually looked like structurally.
Right. For years, there was this ongoing mystery. We knew the basic components, TERT and HTR, and some associated proteins. Early clues hinted it might be a larger complex than just one TERT and one HTR.
Hints like what?
Well, biochemical studies, things like cross-linking, where you chemically link nearby molecules, or FRET experiments measuring distances, they suggested interactions that pointed towards maybe two units coming together, a dimer.
But the picture didn't show that initially.
Oh,
You
wow.
mentioned cryo-EM before.
Correct. The initial high-resolution structures we got from cryo-electron microscopy, which is like this incredibly powerful microscope for seeing molecules, those images mostly showed what people called the monomer model.
Meaning just one unit.
Yeah, one single telomerase unit. It looked kind of bilobed with the main catalytic part, the engine, and then another section called the ACA RNP lobe, which seemed important for structure and maybe recruiting the enzyme.
So the pictures pointed one way, but these other experiments hinted at something else.
Precisely. Even the way certain mutations that cause diseases clustered together seemed more consistent with the dimer structure. It created this significant knowledge gap. Was it really just a monomer? Was the dimer real? If so, what did it look like?
So that lingering question, that's what these researchers set out to answer.
Exactly. They decided to tackle this head-on, and their approach was, I think, really smart.
How so?
Instead of just purifying telomerase and assuming it was all one thing, they collected huge amounts of cryo-EM data from their samples and specifically looked for differences for architectural diversity.
They were actively searching for other forms, not just trying to refine the picture of the monomer.
Right. They were open to the possibility that maybe the sample wasn't uniform. Maybe there was something else in there, even if it was rare.
And they didn't just rely on the pictures, right? You mentioned validation.
Absolutely crucial. Getting a structure is one thing, proving it's biologically real and meaningful is another. They did a whole battery of tests.
Like mutagenesis? Changing bits of the enzyme?
Yes, making specific changes to parts they thought were important based on the structure, then doing biochemical assays to see, okay, does messing up this connection actually affect how well the enzyme works?
And they look in actual cells, too.
They did, in-cell studies to confirm that the structural findings translated into real effects on telomere maintenance inside a living system. It's that multi-pronged approach that makes the conclusions so robust.
Okay, so what was the big reveal? What did they find in that diverse sample?
They found it. From a small but distinct subset of all the particle images they analyzed, they successfully resolved the high-resolution cryo-EM structure of a human telomerase dimer.
So definitive proof, not just hints anymore.
Definitive structural confirmation. They could see it clear as day. The 25-year-old mystery was, in large part, solved.
Wow, so what does this dimer actually look like?
It's quite intricate. It's a large assembly, 26 subunits in total. Essentially, it's formed by two complete telomerase monomers, each with its own catalytic core and its own ACA RNP scaffold.
And they're intertwined somehow.
They are, yes, intertwined. Though it's important to mention, as they noted in their purified samples, this dimer form was present, but it was less abundant than the monomer. The monomer is still the predominant form they saw.
Okay, so it exists, but maybe it's transient or less common in those specific conditions. Now, here's what I find fascinating. How do these two units interact? You'd think maybe the engine parts, the catalytic cores, would work together somehow.
That's what some people might have expected, maybe some kind of cooperative action. But surprisingly, the researchers observed no direct interactions between the two catalytic cores at all.
None. So they're both just doing their own thing.
It seems that way based on the structure and their biochemical tests. Both catalytic cores appear to be active, capable of engaging with telomeric DNA independently. No signs of positive or negative cooperation between them in this dimeric structure.
Okay, so if the working parts, the TERT cores, aren't directly interacting, how are these two units holding hands? What's the connection?
That's the key finding. The dimerization, the connection, is primarily mediated by the other part of the enzyme, the ACA RNPs.
The structural scaffolding part.
Exactly. Remember, each monomer has that ACA RNP lobe. They found specific interactions where the telomerase RNA, the HTR, from one monomer makes contact with the HACA proteins of the other monomer.
So they're linked to their structural components, not their active sites.
Precisely. It's an interaction across the HACA domains.
And this isn't just a minor structural detail, right? You mentioned a link to disease.
A crucial link. They found that several mutations known to cause premature aging diseases, like dyskeratosis congenita,
Which is characterized by really short telomeres.
exactly. Those disease-causing mutations clustered directly at this newly identified ACA-mediated dimerization interface.
Wow. Okay, so that immediately suggests this interface is functionally critical. What happens if you mess it up?
They tested that directly. When they introduced mutations designed to disrupt this HACA interface, it significantly impaired the proper assembly of the whole telomerase complex.
Assembly is affected. What
Right.
about its actual job? Extending telomeres.
That was affected too. Disrupting the interface reduced the enzyme's catalytic activity and compromised its ability to maintain telomeres in cells.
So this directly explains the short telomere problem in those diseases, because the telomerase can't form this dimer properly or it falls apart.
That seems to be the direct implication. The dimer interface is essential for proper assembly and full function. Without it, you get dysfunctional telomerase, short telomeres, and disease.
So what's the thinking now on why it forms this dimer? If the catalytic parts work independently, what's the point of dimerization?
The researchers propose a really interesting functional role. They think the dimer likely serves as a critical assembly intermediate.
Meaning it needs to form the dimer first to get built correctly.
Kind of. The idea is that forming the dimer helps to stabilize the HACA proteins and the RNA as the whole complex is being put together. It's like a necessary step to ensure everything folds and assembles correctly before the enzyme fully matures and becomes active, potentially even before it splits back into monomers, or maybe some function requires the dimer itself.
That makes sense, like a quality control step during construction.
Something like that. And they even drilled down into finer details. They identified a specific HACA protein, GAR1, as a key player.
How does GAR1 help?
It seems to facilitate the dimerization through specific GAR1 interactions across the two halves of the dimer. It also binds to another protein complex involved in structure.
Okay, and there was something else unexpected found within the structure too, something about the RNA.
Yes, quite interesting. They found a specific structure called a G quadruplex formed at one end of the telomerase RNA, HTR, within a single protomer, a single unit. And GAR1 seems to help stabilize this structure too.
So GAR1 is like a linchpin, involved in both stabilizing the RNA structure and mediating the dimerization.
It seems to be playing multiple important roles in holding everything together correctly. It's a really complex network of interactions they've uncovered.
This really does feel like a game changer. I mean, first off, just solving that 25-year-old mystery about whether the dimer exists and what it looks like, that's huge.
It absolutely is. Scientists have been debating this for decades, and now we have this clear, high-resolution picture. It provides a solid foundation.
And the way they got there, the methods sound pretty cutting edge.
Oh, definitely. They developed this really sophisticated pipeline just to produce and purify the entire human telomerase complex. Not just bits and pieces, but TERT, the full RNA, all the key HACA proteins, even a piece of telomeric DNA bound to it.
Getting all those parts together and stable must have been a massive challenge.
Immense. And then the imaging itself using cryo-EM with special graphene oxide grids and a technique called cryofiber thinning to handle these fragile samples. That was state-of-the-art.
Allowing them to get that high resolution, you said 3.2 angstroms.
Yes, which is approaching atomic detail. It lets you see precisely how the different parts fit together.
You mentioned something about a classification-guided workflow. How did that help find the dimer?
Right, so cryo-EM generates hundreds of thousands, sometimes millions, of particle images. This workflow is a computational way to sort through all those images and group together the ones that look similar.
So it allowed them to computationally separate the images of the dimer from the more common monomer images.
Exactly. It was key to teasing out that lower abundance dimer state and revealing its unique structure, including that scissor hinge interface mediated by the HACA components, something totally missing from the monomer models.
And then they backed it all up with those functional tests, the mutation swaps and the single molecule assays.
Yes, those were critical, showing that if you break the connections they saw in the dimer structure, you literally cut the enzyme's processivity, its ability to add multiple repeats in half. That's direct functional proof.
So this isn't just a slightly better picture, it's a leap in understanding the whole machine.
A massive leap in resolution and completeness compared to earlier work. And seeing the whole thing in such detail opens up completely new possibilities.
Like what?
Well, for computational work, like in silico drug design, you can now computationally screen for molecules that might bind specifically to pockets in this dimer interface, for instance.
Targeting the dimerization itself.
Potentially, yes. A level of targeted drug discovery that just wasn't feasible with the lower resolution or partial structures we had before.
It also sounds like a great example of scientific collaboration.
Absolutely. The paper acknowledges leveraging resources from major cryo-EM facilities, mass spectrometry experts, RNA chemistry groups. It was a real multidisciplinary team effort.
Bringing together structural biology, biochemistry, chemistry, computation.
You need all those pieces to tackle something this complex, and they highlighted the importance of data sharing and using tools like AlphaFold to help build the models, which sped things up.
Were there key individual contributions mentioned? Sometimes specific breakthroughs hinge on one person's work.
They did mention a few. Sebastian Balch did heroic work on the co-expression system and optimizing the samples for imaging that's often a bottleneck. Zala Sekne used specialized cross-linking mass spectrometry to actually pinpoint where on the proteins the interactions were happening, confirming the HACA interface.
Providing that chemical evidence alongside the structural picture.
Exactly, and the senior author, Thi Hoang Nguyen, apparently made the crucial decision early on to go for the full-length RNP, the whole complex, instead of taking shortcuts with easier-to-handle truncated versions. That commitment to the complete system was likely key.
Sometimes the harder path is the one that leads to the real breakthrough.
Often the case in science. They also mentioned how cross-training within the team, people learning techniques outside their core expertise, really helps speed up the cycles of experiment and analysis.
So the bottom line is, this paper fundamentally shifts the paradigm. We have to think about telomerase differently now.
I think so, yes. It moves us from mainly thinking about a monomer to understanding that this dimer state exists and is functionally important, likely for assembly and maybe other roles too. It redefines potential regulatory sites.
And it reframes how we interpret those disease mutations.
Right. It suggests many of them might act by dimer uncoupling, preventing this crucial dimerization or destabilizing it, rather than just affecting the catalytic site directly.
Okay, let's talk about the future then. What are the big therapeutic implications here?
Well, they're potentially huge. On the cancer front, this dimer interface presents a completely new target, a new Achilles heel.
Instead of just targeting the active site, which has been the main approach, you could develop drugs that specifically block the dimerization.
Exactly. Drugs that prevent telomerase from assembling or functioning properly by disrupting this interface. That's a whole new strategy for cancer inhibitors.
And what about for aging or diseases caused by too little telomerase activity?
On the flip side, understanding this dimerization gives us a new handle, potentially, for therapies aimed at boosting telomerase function or stability, maybe finding ways to promote or stabilize the dimer for telomere extension gene therapies.
So new ways to both inhibit and potentially enhance its function by targeting this assembly process.
That's the exciting prospect. Of course, we need to be realistic. There are still limitations.
Like what?
Well, this is still primarily based on studies of purified enzyme outside the cell. We really need in-cell structural validation to confirm this dimer exists and looks the same within the complex environment of a living cell.
Things might be different inside a cell.
They might be. There could be other proteins, other factors inside the cell that interact with telomerase and maybe modulate this dimerization in ways we haven't seen yet in vitro. So it's a massive step forward, a landmark study for sure, charting a new course, but it's not the absolute final word. More work needed.
Okay, so let's try and recap the big aha moment here. This enzyme, telomerase, which is so fundamental to our biology, tied up in aging, tied up in cancer. For the longest time, we thought of it as a lone wolf. But this new work shows it's actually this dynamic intertwined dimer, two units linked together.
And crucially, that linkage, that dimerization through the HACA components is absolutely key for its proper assembly and function.
And disrupting that very linkage is directly connected to serious human diseases.
That's the core finding. Structure linked directly to function and disease through this dimerization interface.
Which brings us to our final provocative thought for you, the listener, to mull over.
Okay, consider this. If the proper assembly of this complex telomerase dimer is so critical for keeping our telomeres healthy,
Right.
how might future therapies evolve? Could we move beyond just trying to switch telomerase on or off?
You mean focus instead on correcting its assembly process?
Exactly. What if we could develop therapies, maybe new kinds of small molecules, maybe sophisticated gene therapies that don't just target the enzyme's activity, but target these specific assembly pathways? Could we ensure it forms correctly in aging diseases or disrupt its formation selectively in cancer?
Targeting the construction process itself rather than just the final product. That definitely offers a whole new angle to think about for treating these fundamental diseases.
It's a fascinating possibility opened up by understanding this new layer of complexity in telomerase biology.